Hello, biology students. So we're going to be doing a gene expression review here. This is the review that I gave you in class. Well, I guess not technically in class. In virtual school as we were preparing for our test. So each of these is a possible test question on your assessment coming up. So I offer all of these as practice and examples for down the road. Okay. So keep in mind, we're going to be using our genetic code table. So if you have that printed out, go ahead and get that out. I have mine out, and we'll use it to decode some of these problems. So the first one, the most sort of basic thing I could ask you is to transcribe the following DNA, right? So we have a DNA sequence there. How do we know that it's DNA? It has the T's there. So what you would do is simply convert this DNA code into an RNA code using complementary RNA nucleotides. The trick to that is every time you see an A, you need to fight the inclination to put a T. So instead we need to put a U. I don't mind if on an activity like this, you even just go find all the A's and put the U's, since that's what's going to be most likely the challenge. And I've seen this in previous work as we've gone through. Keep in mind, if you see T's in your RNAs, you're incorrect. So now we can go ahead and just reverse complement everything that we see there. So your sequence for that would be there in the red. Okay, so no T's in your RNA. We got to stop that habit. Translate the following. Okay, so again, some of the problems that I'm seeing is I'm seeing students retranscribe this. So keep in mind, translation is the process of converting these using this code into a chain of amino acids. Okay, so you should end up with a chain of amino acids. It might look something like PRL, for example, if you're looking at the single letter abbreviation. Okay, here's some of the stuff that I'm seeing though, which is somewhat problematic. When you quote retranscribe, basically what students are doing is they're taking their DNA code and they're just converting it back into a different RNA. So students are putting U, A, C, G. That's not what translation is, okay? So keep that in mind. That is an incorrect response. So let's cover a correct response. So basically the first thing you have to do is you have to break this up into codons. Okay, so I have 5 codons there. Now I need to go to my genetic code table. You hear that? It's right here. So there's no need to memorize this. In fact, I've put a copy of it in the background of all these slides, but it's pretty handy dandy to have it next to you. So if I'm looking at my code and I see AUG, I know that that's going to be an M, and we know that all proteins have that because the AUG is the start codon, so methionine is found at the beginning of all these proteins. So then I'm looking at CGC. I go to my genetic code table. That is arginine. CUA is leucine. UAC is tyrosine, which is the abbreviation Y. And CGA is another arginine. So whenever you translate, amino acids need to be the product, not a complementary RNA sequence. Moving along, what DNA sequence creates the RNA sequence? UAC CGA. So, when we go over this in our lessons, I teach you that gene expression is basically one step forward, transcription to translation. DNA to RNA would be one step forward, transcription, and then taking that transcript, translating it into amino acids. So there's 2 steps in gene expression. Here, I'm asking you to go one step backwards to show me how the process works to go back to that DNA. So I don't care where you put this sequence, you need to tell me, given this RNA here, what was the DNA that created it, okay? So we know that where there's an A, creates a U in RNA. So T, G, C, C, T, T, A, A. that would be the code. Now, if you want to practice it like you do in math, if you solve for a variable, why not put it into the equation, see if it works? So if this is my DNA now, I should be able to get RNA that is the same as our starting sequence. So if I do that, again, fight the urge to put T's here, U, A, C, G, G, A, U. And if I'm looking at this here, that is correct, that is correct, that is correct, that is correct, 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 correct. Okay, so that would be a 1 step backward. You might get on the test 2 steps backwards. I'm just trying to see if you understand the process of gene expression. Okay, give a codon that codes for glycine. So the single letter abbreviation for glycine is G. So we're going to find where all the G's are. So here, G, G, and I sometimes note it with an X. GGX gives you glycine. So any RNA codon that has GG starting with it, any RNA letter afterwards is going to give you glycine. So it could be GGA, could be GGU, could be GGG, could be GGC. So GGX for that one. Give a DNA code that will ultimately create tryptophan. So here we have to find where tryptophan is. So tryptophan is right here next to this stop. There it is. So the RNA for tryptophan is UGG. Now if I'm trying to convert back to DNA, basically this 2 steps backwards, I know that ACC would give you UGG when transcribed. Okay. So your answer for a DNA code that ultimately gives you tryptophan, noted by the W, is ACC. And I have to tell you, I really like this question because there's only one answer to it. If you note here, there's only one code for tryptophan on this genetic code table. What amino acid is produced by TAA? Well, you didn't give us enough information, did you? Well, actually I did. If you see a molecule, a DNA molecule that has TAA in it, you know you have to go 2 steps forward. You have to perform all of gene expression. So you have to transcribe and translate. You know that it's DNA because it has a T in it. Okay, so there's your DNA. Your RNA would be AUU. And then if we go to amino acid after translation, you would see on your genetic code table, AUU gives you isoleucine. So the answer there would be isoleucine. Give a possible DNA code for this chain. Okay, wow. So I really like this question too because it is very, 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 very unlikely that you guys would be able to come up with the same DNA sequence for 4 continuous amino acids. Okay, so keep that in mind as you're doing this. So I'm going to say this is the amino acid. I want to go down here to make the RNA. Here I want to go here to make the DNA. So we're going, again, 2 steps backwards. So you find valine here. Valine can be GU anything. So I'm going to say my RNA for this valine is GUU. Okay. Now I know that GUU is coded for by CAA in DNA. Okay. Now for serine, I'll stay right here with serine and say UCC. So UCC for my RNA, which would be AGG for my DNA. For cysteine, right next to it, I'm going to say UGU. UGU for my RNA, which would be ACA for my DNA. And last but not least, arginine, go with this side over here. I'm going to say CGG RNA. CGG RNA, which would be GCC in your DNA code. So that's your DNA code. Now again, you can double check it by seeing if you get this RNA product through transcription, and just cruising through quickly, it looks like that's correct. And then you take your RNA code and use your genetic code table to double check. GUU is valine, UCC is serine, UGU is cysteine, and CGC is arginine. Okay, so the fact that there are several ways to get these will lead to a highly variable DNA code. Keep that in mind. Okay, next one. Show full gene expression from DNA to protein, chain of amino acids, of a 15 base long DNA sequence of your choosing. Okay, so there's a couple of approaches to this. You can just write out a long DNA sequence and hope that you don't make a stop codon. Or you can kind of be clever about it and avoid a certain quadrant, that being the quadrant where all the stops are. That is the ones that start with U. So if that's the case, I would want to avoid A's as my starting letter for my DNA sequence. Okay, so because there are 15 bases, that means there's going to be 5 codons. And I know that I don't want to put A at the beginning of any of them. So I'm going to put either T's, G's, or C's at the beginning. And then whatever you put next after that is it's not really going to matter. So okay, so there's my DNA. Now I need to transcribe it into RNA. So we put the complementary RNA nucleotide there. So I think that matches up. The thing you want to double check is anytime you see an A in your DNA, make sure that you've paired it with a U in your RNA, and that looks fairly correct to me. So then my amino acids, use my genetic code table. What does AGG give me? That's going to be arginine. What does CUA give me? That's going to be a leucine. GUA is going to be a valine. AUC, that's going to give me an isoleucine, and GGG is going to give me a glycine. We did it. Again, this is a highly variable question. Each of you should be able to come up with a different answer on your own. If your codon is AUA, what is the anticodon of the tRNA that brought the amino acid? So again, if we're looking at RNA, and this being messenger RNA, that's what's going to give you the codon. So I'm asking you if it's AUA, what will be the tRNA? I'll put anti-codon, alpha codon. So you just do complementary RNA nucleotides. So in this case, it would be UAU. Okay. Keep in mind, though, this is the one you use on this genetic code table to determine the amino acid that's being brought. So AUA would bring isoleucine. So this tRNA would be bringing isoleucine. And that's what we get to here. So if you are a tRNA, messenger RNA, if your tRNA has the anticodon GCA, that means that the messenger RNA was CGU. We use that codon CGU to say that this tRNA right here is carrying, what do we say, CGU? CGU is arginine. So it's carrying an arginine. Okay, now we get into a little bit of the mutations. I'm sticking with substitution mutations here, but I'll also ask about insertions and deletions. And I want you to show me the different effects that can occur because of a substitution mutation. Okay, so in this case, the substitution mutation, I want you to see show me a silent effect. So silent effect means the same amino acid is produced. So let's go through this before the mutation. I'm going to say before the mutation, you have RNA that is GCG. If I go to my genetic code table, GCG gives me alanine. So this needs to be the same thing here. Okay, so if you look at a genetic code table, and I didn't have space to put one on this slide, what you're likely to see is some flexibility at that third RNA nucleotide position, but little flexibility at the first position. So for our DNA, it's a pretty safe bet that we don't want to change that first DNA nucleotide to keep it silent. There's a good chance too that we don't want to change the second one. Okay, so if you look at your genetic code table, CG, what I say usually anything, will give you, sorry, back it up a second. What am I doing? Sorry, we got to transcribe this thing. So this would be GC anything. GC anything would still give you alanine. So it doesn't matter, you know, if you want your RNA here to be C, that would make this a G. We see here that you have a substitution mutation compared to the original. Instead of getting the G, you get a C, but it doesn't matter because in both cases you get an alanine. Sorry about that, guys. That threw me off for a minute there. But you see the point there. They both result in a silent mutation, silent effect. Okay, in this case, we want to get to what happens when you perform a substitution mutation that results in a missense effect. Now remember, missense basically means you get one amino acid that you should have gotten and it changes to a different amino acid. So here you're going to get this different one here. Okay, so the DNA that we're working with, and I should have filled this out. Let me pause this and fill this in for you. So that'll speed up the process. Now I have some information for you. So if you look at your DNA sequence here, it starts off with ACC, which transcribes into UGG. If you look at your genetic code table, UGG gives you tryptophan. So if we want to show a missense effect, you have to change one of the DNA nucleotides such that it results in a different amino acid once gene expression is performed. Okay, so there are two ways that you can do this pretty easily. If you keep the first two DNA letters intact, which will give you UG, you can change the last one two different ways. So here's one solution. If you change that original C to a G, that'll give you UGC, and UGC is going to give you cysteine. Okay. Similarly, if you change that last letter to an A, which gives you UGU, UGU also gives you a cysteine. So there's two different ways to make a missense with the genetic code for tryptophan to make cysteine. Last but not least, I want to do a substitution mutation that leads to a nonsense effect. So this nonsense mutation or effect is going to result in a stop. Okay, so here we have the DNA sequence ATG, which gives you UAC. That's tyrosine. Now if I change one of these last letters, again, if I change it to ultimately getting to a UAA or a UAG, that's going to give me, in both cases, a stop. So the DNA that would give you that is ATT. So I've changed this G to a T here, or ATC. Again, I change that G over here into a C over here, and that transcribes into UAA and UAG, which both give you stops. So the protein would stop being produced at that point. Okay, so there it is. Your test questions will be from these 14 different examples. Hope that helps. See you next time.